Okay, welcome to our last tutorial session, tutorial 4. I did question 2, so let's just dive into it. It says physical maps are based on DNA sequence based on markers that can be associated with specific genes or phenotypes. One technique is creating physical maps that rely on restriction mapping, and where the DNA is cut with the restriction enzyme, endonucleases that recognize specific short sequences. Since the restriction enzyme are usually based on those original harvested from microorganisms, they have names such as EcoR1. Eco stands for I can't really pronounce that, but yeah, you can see this. So imagine you have a linear piece that is 14 kilobase pairs long. To make restriction map, you first cut with EcoR1, then SMA1 alone, and finally both. So one thing to notice when we look at this data is that all of these should add up to 14. So 3, 5, 6, 14. 7 plus 7 is 14. 2 plus 3 plus 4 plus 5 is 14. And how we can determine these cuts. Okay, actually, let's just dive into question A. How would you have gone about determining how large each fragment of DNA was if you were doing this experiment? So, for testing how large DNA fragments are, the most popular way to do this is through gel electrophoresis. And so after we treat with our restriction enzymes, we could run a gel. So this is just an example. This isn't actually the gel for these two different ones, but how this works is that you have shorter length pieces on the gel closer to these wells down here. So let's say we had our eco restriction enzyme put in a well and had different cuts. The shorter pieces would be down here and the longer ones would be up here. Then we have a DNA ladder that we can compare it to determine and approximate how many base pairs long these would be. So I'll get more into this. This is just kind of a rough diagram, but that's essentially what happens. Once you run it, these base pairs will move and they'll show these DNA bands, which we compare to the ladder. And so that's how we would determine the size of each fragment. So for B, we have representing your original 14 kilobase DNA as a line. Draw the relative positions of the SMA1 cut site. So this one's simple enough. Over here we see that there's 7,000 base pairs. So there's just one cut that's happening here. There's two sections, and easy enough. 14, half of 14 is 7, so it's going to be right in the middle, and that's our SMA1 cut site. Now, for B, it asks us to show the cut sites, or all the potential cut sites for EcoR1. So now we have two cut sites that we could work with, and these are each going to be 3,000, 5,000, 6,000 kilobase pairs, so 1,000 base pairs. And here I have all these options here. So basically, one of our options could be we have a cut here, 3,000, and then we have 5,000 and 6,000 base pairs. Or we could have 5,000, 3,000, 6,000 here, or 5, 6, 3. And then these ones pretty much, I'm leaving the middle the same for these, but then just switching each side. So realistically, it's between these three, and depending on how you want to read it, left to right. Because if we go down to, so here, sorry, 1, 2, 3. So now we go here, we have 5,000 in the middle, but instead we have 6,000 over here, 3,000 over here, 3,000 in the middle, same over here, but we're switching these, so 6,000, 5,000, and 6,000 in the middle, and we switch those. So that just is able to show us all the potential possibilities reading it from left to right. So yeah, those are our cut sites for ECO1. Now, what do we have next? Draw the actual restriction map with distance and kilobase pairs on your digestion with both. So basically what we're going to do now is now I have all these options of ECO1, right? And we have only one option here for SM1. So basically what we're going to do is we're going to take this 7,000 and we're going to see if this makes sense. We're going to apply it to all of these lines and figure out which line makes sense with this over here. With both, we need to have 2,000, 3,000, 4,000, and 5,000 kilobase pairs. So let's start with our first line here. So we have 3,000, 5,000, 6,000. So if we were to go like this, and so let's say that's 3, so imagine from here there's another 4,000 base pairs. Would this make sense to make a cut there? Would that give us our desired product? Well, no, because we still have a 6,000 base pair over here, and as it says over here, there's no 6, it's just 2, 3, 4, and 5. So that we can cancel out. That's not going to work. Now, we can go to our next one. Same deal, pretend, so we have 5. Now let's add another 2,000 over here, right? Let's say that's our cut. Will that work? No, because that leaves us with 1,000 kilobase pairs right here. 5 and 2,000 here, and 6,000 again. So that does not make sense with 2, 3, 4, and 5. Okay, so now let's try this one. Start from left to right. 5,000, add another 2,000, put that here. Let's mark that. All right, this looks a little promising. We have 5, we have 2, oops, sorry, that's a 3, 2,000 kilobase pairs. 6 minus 2 would be, this would make this 4,000 kilobase, and then 3. So there we have it. We have our cut right here. That'll give us 5, 2, 4, and 3. And then it's the same idea down here. I just starred these. It's just the different order. But yeah, if we were to go over here, we just know that has to start from the 5,000. So 7,000 over. Add another 2,000 there. That gives us 5, 2, 6 minus 2. That gives us 4 and 3. So now I drew it out over here a little more neatly. For D, this is what it should look like. So at 5,000 we have our Eco 1 cut. Then we have our SMA 1 cut there, leaving us with 2,000 kilobase pairs. Then we have our another Eco 1 cut. So then we have 4,000 and 3,000 kilobase pairs. Hopefully that makes sense. Now, imagine, okay, here we go. Imagine you have a gene M+ that contains the SMA site, and when the site is mutated M-, SMA can no longer cut, and the mutation causes an albino phenotype in the homozygous condition. Draw an electrophoresis gel that shows restriction fragments for a double digest in all wild type and albino conditions. So essentially what this is saying is that in albino conditions, this guy right here, SMA1, cannot cut. So since this cannot cut in the albino conditions, it will show a different banding pattern on a gel, electrophoresis gel. So here, I drew this over here. This will explain it a little bit better than I did earlier, than this kind of rudimentary looking guy. So yeah, basically how gel electrophoresis works, anyways, just a quick little overview, is that shorter bands can travel further, and as we know, DNA is negatively charged. So, and then it will move along this gel towards the positively charged end. So shorter bands can move further along, and the longer bands kind of get stuck, so to say, in the gel at these different positions. So with our wild type strand, we can assume that there'll be bands at 5,000, 2,000, 4,000, 3,000 base pairs. And what we use is a DNA ladder, and this is something with known distances of the gel that can allow us to estimate how many base pairs there are. So in a perfect world, after doing this experiment, we should have a band at 5,000 right here, 4,000 right there, 3,000 and 2000. That's going to be our wild type gel. However, for the albino guy, we cannot cut at this site. So that basically brings us to our numbers over here. This isn't working, so we can only expect bands at 3, 5,000, 6,000 kilobase pairs in length. So it should look like this. We'll have a 6,000 band, a 5,000 band, and a 3,000 band. Alright, hopefully that makes sense. Lastly, said what if there's a gene L plus, that when mutated, L minus, the ECO1 restriction enzyme can no longer cut at both the sites, and when it's mutated, let's call it cystic fibrosis. I don't really know if cystic fibrosis has anything to do with ECO1, but this is just a hypothetical. Yeah, this is just kind of going to reiterate what we're talking about up here. So now, in this case, I'm going to write SCF there. Our gel should look like this. We have our ladder again. Our wild type should emulate this up here because it's running smoothly. We have our 5, 4, 3, and 2,000 base paired, but with this cystic fibrosis DNA, ECO1 can't cut, so that leaves us with only one cut at the SMA site, and that's at 7,000 kilobase pairs. Yeah, and then I also said, this is just kind of like a discussion question, how could you determine another restricted enzyme that could make more cuts in cystic fibrosis sample? It's kind of like a broad idea, but what I decided to do is say, what you could do is you take this DNA, you run a Sanger sequence, you find the actual DNA sequence of that 14,000 base pairs long, and then through analysis you could find other types of restriction enzymes that would have matches in that sequence that would make more cuts, because basically how these guys work is they bind and cut at certain sequence sites. Like, let's say there was a restriction enzyme and it liked, so it was like CAT or CTA-T, and when it saw that, it recognized it and cut it at that site. So yeah, that was just a quick little discussion on that, and that's what I would do. And hopefully that all makes sense. And yeah, have a good one. Bye.